My name is Adrian Lawrence. My team and I performed a retrospective evaluation of SMOF lipids on liver function for home parenteral nutrition patients. Those who worked on this project have nothing to disclose. This is a copy of my poster. There are currently several different FDA-approved formulations for intravenous lipid emulsions available in the United States that differ in the fatty acid content and composition of the emulsion, which can include soybean oil, medium-chain triglycerides, olive oil, and fish oil. Soybean oil-based lipids were the main source of intravenous lipid emulsion until SMOF lipid was approved for use in July of 2016. Potential complications following the use of soybean oil-based emulsions include elevated bilirubin levels, intestinal failure-associated liver disease, and cholestasis. Due to the safety profile of soybean oil soil-based emulsions, other alternatives have been investigated. One of the alternative intravenous lipid emulsions contains soybean oil, MCT, olive oil, and fish oil, more commonly known as SMOF, which provides a combination thought to have beneficial biological and physiological effects. Currently, data for usage of SMOF in adult populations is scarce, though the product is considered safe and has been used in Europe since 2007. Thus, more clinical evidence is needed concerning the long-term effects. The goal of this study seeks to assess the safety and tolerance of SMOF in comparison to soybean oil emulsions in adult patients receiving parenteral nutrition at home. The primary objective of this study was to compare the effects on total bilirubin before and after the patient switches between the two products. The secondary objective was to determine and compare the effects on the inflammatory markers and liver function panel. This study was an IRB-approved single-center retrospective chart review conducted from May 1, 2016 through November 30, 2018. All patients served as their own control. The study was separated into two phases based on the lipid emulsion being administered. The patients were on soybean oil emulsion first for at least 8 weeks before transitioning to SMOF. We included all home patients regardless of diagnosis or route of administration. Patients were included if they were greater than 18 years of age, met the lab requirements for each phase, and were at home for at least 2 months post-initial discharge. Patients were excluded if they did not receive lipids at least 3 times per week due to insufficient amount of the product received for evaluation. A total of 184 patient charts were reviewed. 158 of these patients were excluded, resulting in 26 patients being included in the study. For the baseline demographics, there were no dose differences between the dosing for patients on soybean oil versus SMOF, and the patients had complex diagnoses and indications, with the majority of our patients having SBS or short bowel syndrome and fistulas. With these diagnoses, it is understandable that the patients would necessitate long-term home parenteral nutrition administration. The effect on total bilirubin was evaluated for both of the phases. This was found to be statistically significant with a p-value of only 0.04. The average for the soybean oil group was 2.47 milligrams per deciliter, and the average for the SMOF phase was 1.69 milligrams per deciliter. The next slide will provide a better explanation of the trends observed during both of these phases. This graph demonstrates how the average bilirubin fluctuated throughout the administration of both lipid emulsion products. The y-axis contains the reference range for bilirubin measured in milligrams per deciliter, while the x-axis contains the time period when the averages were obtained leading up to the transition period. By initiating the SMOF product, the curve begins to decrease as it approaches the endpoint of the study duration at 2 months after the transition. This graph demonstrates the trends of the various secondary outcomes. This graph contains the ALT, ALK-Phos, AST, and triglyceride, all of which show a decrease from the initiation of SMOF to the endpoint. The only one that was found to be statistically significant, though, was the one for ALK-Phos, which is seen with the orange line that has a substantially larger slope. In conclusion, SMOF lipid emulsions were a safe and effective alternative to soybean oil emulsions for home parenteral nutrition patients. Results of this retrospective study showed a statistical difference in the total bilirubin levels in the SMOF phase of the study. Based on our current lab value ranges for the hospital of 0.3 to 1, the SMOF emulsion lowers the bilirubin closer to within range. Switching from soybean oil to SMOF fat emulsion as a major component of home parenteral nutrition was associated with a significant improvement in serum total bilirubin and alkaline phosphatase levels. Larger prospective multi-center studies are needed to confirm the relationship between the use of SMOF in home parenteral nutrition and improved liver function tests over time.